I-click na. You are holding a small plastic tube with some clear liquid on it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? Press forward to continue. Right. If you can see the DNA strands short, medium, and long. If the DNA strands were as big as your shoelaces, you could sort them by hand into groups and measure them. Press forward to continue. But there's but DNA strands are molecules so tiny that you can see them even under most microscopes. Is there any way to sort and measure DNA strands in your tube even though you can see or touch them? There is. This is called gel electrophoresis, pronounced as electrophoresis. Press forward to continue. Scientists use electrophoresis, electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How this works? Press forward to continue. This gel is the filter that you can see the yes. This gel is the filter that sorts the DNA strands. It's like a sponge made of Gel-O with many small holes in it, as you can see in the picture. There are small holes. Press forward to continue. Wait. We place DNA samples into the holes at one end of the gel. Press forward to continue. Electrophoresis is how we push the DNA strands through the gel filter by adding an electrical current. You can see in the virtual lab. You can make the DNA move. Press forward to continue. Wait, loading pa sa Short short strands move through the holes in the gel more quickly than long strands over time. The shorter strands in the sample will move farther away from the starting point than the longer strands. DNA strands of same length will move at the same speed and end up grouped together. In this way, the DNA strands in the sample sort themselves. Press forward to continue. Staining the sorted groups of DNA makes them visible with the naked eye. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. Press forward to continue. Now, it's your turn to run a gel. That's scientist slang for gel electrophoresis. Follow along the steps shown above. Press forward to continue. Step 1. Make the gel. Step 2. Set up the gel apparatus. Step 3. Load the sample into the gel. Step 4. Hook up the electrical current and run the gel. And lastly, step 5. Stain the gel and analyze the results. Press forward to continue. Step 1. Here is that you will need to make a gel. Powdered agarose, buffer, a flask, a microwave, and the gel mold in the gel camp. Making an electrophoresis gel is a lot like making a jello, but you don't want to eat this type of gel. Press forward to continue. Put a small amount of agarose into the flask. Agarose is a dried powder similar to gelatin but made from seaweed. Click and drag the spoon to the flask. That's a- Wait. Add add some buffer to the flask. Yes. We've loosely placed plastic wrap over the top of the flask. To prevent the liquid from boiling over, open the microwave by pressing open button. Open the microwave by pressing open button. Place the flask containing the buffer agarose mixture into the microwave. Heat the microwave until the agarose melts into the buffer. We've moved the plastic wrap from the top of the flask. Pour the melted mixture into the mold. Notice that the mold has a tape on each to hold. Place the cam on the gel at one end. The notches, the notches in the gel mold it into place. Press forward to continue. Let the gel cool and solidify. This usually takes half an hour but will speed up the clock for you. As the gel cools, tiny holes will form in it. Press forward to continue. Carefully remove the cam leaving the empty shells. Yes. Now it's time to set up electrophoresis box. You will need the gel you just made on an electrophoresis box and another bottle of buffer. Press forward to continue. Pour the buffer into the electrophoresis box. Place the gels still in the mold into the electrophoresis box. We have removed the tape from the ends of the gel mold. The gel should be just barely submerged into the buffer. The buffer conducts the electrical current from the one end of the gel to the other. It will also keep the gel from drying out during the experiment. You're ready to load the DNA sample into the gel. Here's what you'll need. Loading buffer, the tube of the DNA, the DNA side standard, a micropipettor, the electrophoresis box containing buffer in the gel. And lastly, you will need pipette tips. Press forward to continue. Okay, okay. Sa taman. Sa taman na kuan, Andre. with a clean pipette pipette, use the micropipette to suck up some loading buffer, then add the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if we try to load it directly into a well. The loading buffer contains a dye that makes the sample too easy. It's also slightly gappy. This makes the DNA sample thicker so that you will drop it into the well instead of floating away. The DNA size standard already contains the buffer. Next, you will use the micropipettor to transfer the DNA sample from the tube into the well of the gel. First, suck up some DNA sample into the pipette tip. Suck up. Next, i- and, and enjoy the DNA sample from the pipe into the first well of the gel. Press forward to continue. Eject. Eject the DNA sample. Yes. Nice job, Char. In real life, loading the DNA samples into the wells takes some practice. So, don't be disappointed if you target the first few times. Press forward to continue. Using a clean tip, use the micropipettor to suck up some DNA standard. Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths running it on the gel. Will give you a reference by which to estimate the length of the DNA strands in your sample. It's time to turn on the electrify and run your gel. When you turn on the power, the black end will generate negative charge. The red end will generate positive charge. Together, they will pass a current through the gel. DNA has a negative charge. To move the DNA through the gel, you must put back the black cord, which is the negative charge closest to the wells. Press forward to continue. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Click the location on the power supply where you want to plug in the black cord. That's right. Now, click the location on the power supply where you want to plug in the red cord. And turn on the power supply. Your gel is off and running. Let's look into the gel box to see what's happening. Check for tiny air bubbles coming out of the electrolytes. So you can see the tiny air bubbles at, at both ends of the electrophoresis box. These bubbles are your proof that a current is running. So the bubbles moving are the proof that yes, the current is moving. Press forward to continue. Repelled by negative charge, the DNA moves through the gel towards the positive charge at the other end. Short DNA strands move through the holes into the gel quickly than long strands. Over time, the shorter DNA strands will migrate farther away from the starting point than the longer strands. We can't actually see the migrating DNA bands which are gray, but we can see them blue dye from the loading buffer as it migrates. Press forward to continue. You've you have finished running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. This time has come to analyze your sample and estimate the length of the DNA strands. Press forward to continue. First, you need to stain the DNA in your gel using the DNA staining solution. This stain is a chemical called ethidium bromide, which binds to DNA and shows up under a fluorescent light. Although we can't see single DNA strands, we can see large groups of stained DNA strands. These groups will as show up as bands in the gel. Press forward to continue. Drag the gel out of the mold and put it into the staining solution. Because ethidium bromide binds to DNA, it can damage the DNA in your cells. If you stain a gel in real life, wear gloves and avoid direct contact with the staining solution. It takes half an hour to stain the DNA gel. We'll speed up the clock for you. Oh, remove the gel from the staining solution and place it on the UV or ultraviolet light box. Turn on the UV box or ultraviolet light box. Ultraviolet light from the or UV from the box can damage your eyes. If you do this in real life, be sure to wear protective goggles. Now you can determine the approximate length of the DNA strands in your sample. Compare the bands from the DNA sample with the bands from known length from the DNA size standards. Enter the estimated length in base pairs or BP. From each strand, your DNA sample electrophoresis cannot tell us the exact DNA sample length of DNA. Just a good estimate, so give it your best guess on the DNA size standards. Press forward when you are finished to check your answers. Estimate, kuha na siya. 1,500. Kay 2,000 naman ang next. Oh, kanang pinaka sa first. May label. Mag-label. Oh. DNA size. Kana 6,000 na dira. Pero na may naka araw-araw siya. 6,000. Kana 3,500 kay between sa kuan 3,000 and 4,000. 3,500. Then dira ang kay kuan 1,500 kay between sa 1,000 ug 2,000. So that's it. We have estimated our size, DNA size strands. So to continue, press forward. Okay. All of your estimations are correct. So that's it. We are finished. Oh, we are done. We are done. We are done with the experiment. So, humana. Cut na ba?
Ako
Para
asang
sa may agi.
balin sa ko sa okay. I'm read. You gonna start? So hi ma'am, we are the group 10. And we are going to present to you our answers about the gel electrophoresis. Gel electrophoresis is used to sort DNA strands according to their lengths. And In this question, where do you place the DNA samples on the gel? Our answer is holes at the end of the gel. And in this question, what makes the DNA move? By adding an electrical current, we can make the DNA move. Short strands move through the holes in the gel more quickly than long strands. And short strands move faster than longer strands. 2. Place the steps in correct order. The first step is to make the gel, and the second step is to set up gel apparatus, and the third step is to load DNA sample into the gel, and the fourth step is to hook up the electrical current, and lastly, stain the gel and analyze results. In the Gel Electrophoresis laboratory, follow the steps to make your own gel and answer the questions as you go. What is agarose made from? So, agarose is a dry powder similar to gelatin but made of seaweed. Melted agarose is poured into a gel casting tray. And what is the purpose of the comb? The purpose of the comb is to create the wells. And the black end generates a negative charge and the red end a positive charge. The bubbles in the electrophoresis indicate what? Bubbles are proof that a current is running. Staining the DNA will make it show up under a fluorescent light. And okay, G, what are your estimates for the number of base pair in the three bonds? So the top one is approximately 6,000 base pairs, and the second is approximately 3,500 base pairs, and the third is approximately 1,500 base pairs. And that's all. Thank you so much. Moro to.
Um,